Okay. Cool. So the way we would practically approach this this problem of binning is if we had a small, if we had one organism that we were interested in a sample, we would probably go and attempt to pick that out using manual binning, because it gives you the best handle on what a single organism could do. This scenario could happen if you've looked in your metagenome and found that there is a specific gene that you're interested in, and it is divergent from the ones that you know, and you're curious what organism it's in. Right? In that case, you could run the whole binning pipeline and get all the organisms, or you could specifically say, okay, let me just go and identify that one. That topic we'll not cover today. We will briefly go over the various binning tools that I briefly introduced, and then see how to run them in practice. And from there, we will look at dust tool briefly, and that will conclude what we're talking about. So what we see, what we see here on my screen is, can I make this bigger? Oh, nope. I can make that bigger, perfect. Okay, so what we see on the screen here is I am on our own lab server. This here in blue shows the directory I'm in, and this here in the brackets at the front show that I'm in an Anaconda environment. I'm in an Anaconda environment that's entitled binning demo, because I think it is useful to know to have a consistent environment to do your work in. Now, it so happens to be that this will be shared on the BVCN website. It so happens to be that a few of the good binning tools that we have available, and the dust tool binning aggregator, are all installable through Anaconda and play well together. So by using this command in detail, you can set up an Anaconda environment that has both the binning aggregator dust tool and 4 different pieces of binning software in them. The pieces of binning software are Binsanity, Concoct, Metabut, Metabut 2, and MaxBin 2. Once you have installed this Conda environment, you can then activate it using the Conda activate command, and that gets you to a situation where we are now. Now, before you start with binning, it is important to do a few steps that are required and will basically avoid a world of trouble later. So one of the things that you have to do before you start binning is the simplify FASTA script. This comes with Binsanity, but is both useful for Binsanity and Concoct. What the simplify FASTA script does is, regardless of what your assembler is, because each assembler gives your context a different header, regardless of what your assembler does, it will change your names to contact 1, contact 2, and so on. This is important for both Binsanity and Concoct, and the simplify FASTA script comes with Binsanity. This is why it's useful to have everything in an Anaconda environment, so you can actually use cross-use the different tools that come with the individual binners to help you out in the long run. You have to run this first so that all your, so that your reference names and your read mapping files that will tell you about abundance are all the same. Now, as we said in the lesson, contig binning depends on both sequence abundance across samples, and it represents sequence and on sequence composition. Sequence composition is easily calculated from the contig, so you don't need any other files. However, abundance is not easily calculated from the contigs because you need the mapping file for that. Like most assemblers don't actually come with a mapping file. So for that, you'll need to make your own mapping file, and these have to be sorted and indexed BAM files. Now, that's what this step and this step is for. In this case, this is two separate steps for bowtie 2. The reason I choose bowtie 2 is that it comes with MaxBin. So it is already installed in this specific Anaconda environment, and therefore can be used to easily get to your assembly. So you see that there's two commands for bowtie 2 build and bowtie 2. Bowtie 2 build will build an index. Basically requires you to provide a reference FASTA file and the base name for your bowtie 2 index. And it will then, so say your FASTA file is example.fasta, your bowtie 2 index name could then be example. And it will then append the index extensions after it. Now, once you've done the build of your index, it can be used as a reference for mapping. So bowtie, as many other mappers doesn't map on raw FASTA files. You have to pre-index your FASTA files. Now, once you've done that, you can run bowtie. You'll see there's lots of options when running bowtie, but again, these come with largely with a set of default presets that you can use. Bowtie is one of the older pieces of mapping software and comes with very, very good documentation. And this here gives you the options, and then the index file, which you've just created using bowtie 2. And then either, if you have paired end files, this is the format. So two different files, you have to specify with a flag -1 or -2. If you have unpaired reads, there's the -u flag. And if you have your paired reads in one file, there's the interleaved flag. This BAM flag is if your reads are in a BAM file, so already an alignment file. This here is important to specify. This is the output. If you don't specify the -s flag with a SUM file, SUM is your sequence alignment file, you will map to standard out. So you'll map to your console, which is not great. So it is important to specify that it writes your alignment to a file rather than just to your output. Now, once you've done the mapping, you have a SUM file. The SUM file is an alignment file, but it's a text file. And most pieces of software like to work with binary files, as well as binary files that are shortened and indexed. So for this requires you to use your utility SUM tools. SUM tools, as the name says, is a set of tools that allow you to play with sequence alignment formats. Lots of these are initially made for eukaryotes, human alignment. That's not what that does. And this allows you to move things from, say, a BAM to a FASTQ, but the ones we're interested in is the sort one, which sorts the alignment by position, and then the view one, which allows you to sort of counterintuitively interconvert between files. So some tools sort, and some tools view. Some tools sort basically give it tells you to sort your sum file, and then some tools view converts it to some tools view converted to a BOM file. Some tools sort allows you to sort your BOM file, and then there's some tools index to finally index your alignment. Those three things, I forgot the third one. Those three things are necessary to get to a set of sorted and indexed files. Now ignore this. What that will look like is something like this, where you have a set of BOM files that are then also sorted and indexed. And once you get to this level, this is where you, this is then where you can, actually start your binning. So most binners will require you to have your sorted and indexed BOM files, and then from there can do something. So if we look at this directory here, we see that that I've put README files for the various binners here, and we'll look at the Binsanity README first. Binsanity README is by far the longest one, because I've mostly taken this from the excellent documentation that currently resides on the internet. Most tools are not this well documented. So for most tools, it pays off to have a look at the README files that come with the documents. So this here, but for this specific one, this comes from this web address. It actually tells you you'll run into trouble if you don't run the simplify FASTA file over here. So if your contigs, even if your contigs are just named with a numeral, you'll run into trouble there. So you have to get to a format that looks like that. Then Binsanity comes with a tool that's called Binsanity Profile, and allows you to take your sorted indexed BOM files and create something that looks like this. So this is your abundance matrix, where for each contig, you have one value per sample. That is your abundance. These are tab delimited. And this one is, Binsanity creates two different ones. It creates a transformed file, and it creates a raw file. Now the raw file is, Binsanity Profile creates these two things. The raw file is useful because you could use that in other pieces of software. The log transformed one is what Binsanity prefers as input. And then using that, we now have this matrix over here, which we can then use to run the Binsanity command. The specific requirement for the various abundance files differs. So for MaxBin, for example, we get an abundance file that looks very similar to what the Binsanity file looks like, except rather than having a column for each sample, MaxBin expects a separate file for each sample. So to run MaxBin, what you need to do is you need to make a specific tab delimited file for each individual sample, and then either give it a comma delimited list as an argument for running it, or give it a file that lists, like here, lists the abundance list file. Alternatively, MaxBin has a feature that it can generate this abundance for you. So basically we'll do the read mapping and BAM file creation and then generating of abundant file for you. And depending on how you want to mix and match, you could feed MaxBin both an abundance list and a reads list. So it is, you have a mix of both. So it's a different way of getting to that same abundance file for MaxBin. Now, then we have the Metabut README, which is basically its standard usage. Metabut comes with two possible ways of running it. It comes with a shell script that wraps the actual program, or the two actual programs that you use, and allows you to feed it a FASTA file and just the BAM files. What they prefer to call the slightly less easy way is to use the script JGI summarize BAM contact depths to make a depth file from your, or make an abundance file from your BAM files. I prefer to run it the second way because it allows me to look at the options of the tools. Whereas this one, you need to know the options of Metabut that you want to use before running it. It is important just to know that this is yet another way to get to the same abundance information. The big difference is that Metabut requires an abundance file which has both the mean and the variance of the coverage of the abundance. And as far as I know, this is one of the few programs that gives you that. So therefore specific to this one. So what you end up doing is you end up creating different different abundance files for each individual binner, even though they use the same information. Now, Concoct is does one thing slightly different than all the other binners. It allows you to split your contigs. So after you've assembled them into long contigs, it requires you to split your contigs into equally sized chunks. So basically saying, okay, we want to weigh every bit of DNA equally well. So in this case, you have, rather than just running a concoct program, there is a there's a step that you start with, which is cutting up your FASTA file, your contig FASTA file into cut up contig FASTA files. And this one will immediately give you a bed file. The bed file is a thing that is borrowed from eukaryotic genomes, and it can tell you where, like, what the stretch of your subsequence is, where a subsequence sits on a larger sequence. What this bed file does is it allows it to take your BAM files, and you don't have to specifically remap all your reads on the cut up contigs that you've made. So now you have, using this secondary helper script, you have you've now made the coverage, and you can actually start running the concoct script. So even though we have 4 different binners and they all require the same type of input, they have 4 different ways of getting to your abundance from your BAM files. And it's important to realize that and to make sure that you prepare these files in between. And then eventually, once you have your abundance files, the binners themselves will run relatively easy. We will not run any of the individual binners today, but rather look at an overview of what Dust tool can do. So in this case, Dust tool is the binning aggregator that is also installed in this Conda environment. And it allows you to aggregate the results from these individual binners. Now, what I'll do here is I'll just run the overview of, or I'll just run it on the sample data that comes with Dust tool. Sample data is not present in the Conda environment, but you can take it from their GitHub. And their sample data looks like this. Their sample data is the contigs, and then the output of 4 different binners. So concoct, maxbin, metabut, which is the 3 we have, and then the 4th one is tetraesum, and then scaffold. They call this the scaffolds to bin file. What that looks like in real life is just, again, a tab delimited file. Well, let's not do cat, let's do head. What? No, well, that's what you get. It's a tab delimited file indicated by the TSV, tab separated, of the scaffolds or contig names or headers without the little caret in the beginning, and then which bin they're in. So in this case, we've looked at the one for concoct, and we see that concoct bin 77 is made up out of all these. So out of all these scaffolds. So it's important to make sure that your samples are in this. Well, this one's from 17, are in this format. To do that, is it this one? Oh, so sad. Okay. To do that, Dust tool comes with a reformatting way, reformatting script, which I'll look up right now. The reformatting script takes your individual bin FASTA files. So if you have, as the output of your bins, you have FASTA files with the contig names in there and grouped with 1 file per bin, as most binners will give you. It will take that, and from those, based on the bin names, will generate this, this file. Now, the way we can look this up, this might be instructive anyway, is we can look at where our Anaconda is installed. In my case, that is over here. And we can look at what the Anaconda environment looks like. This is what it looks like for me. And we'll see that if I look at envs, we'll see a long list of different environments that are present on the server that I work in. However, there is, so my binning demo environment right here is right here. No. However, if we want to see where what is installed in each, or where the packages are installed, we don't go to envs, we go to packages. And then in this case, we'll go to Dust tool, that's 112, and then bin. And this tells you, okay, that's the name. And this tells you which helper scripts are there. The important thing to know here is that the packages are not installed in the environment that they're active in. They're installed in one central location, which means that if you have different Conda environments using the same packages, they are not fully independent of one another. So sometimes there might be crosstalk, which is, I think, the cause of a lot of, or some of the problems that Anaconda users experience. So anyway, this is the FASTA to scaffolds to bin script. Oh, okay, -h. And basically, it requires three things. It requires you to give it a FASTA file, and it requires you to give. It requires it to give you a FASTA extension. So say if your bins are labeled .fasta or .faa or .fsa or .fna, some different programs use different extensions for different FASTA files. An input folder where all your bins are, and those are the only two things. And then that will turn it into a scaffolds to bin file that can then be used in dusttool. And that will be in this format. So this is just a program you will have to run for each of the individual binners. Once you've done that, and you'll get to a format that looks like this, you have the scaffolds to bin files for your different binners and your contigs. You can then run dusttool, and dusttool can be run relatively straightforward in the way that is described in their help message. So in general, it's always a good idea to obsessively use the -h flag to check for what the possible options are. It has a couple of it has two really clean prepared examples. One of these is this one, and you'll immediately notice that dusttool is incredibly wordy. Right? The reason for this is that it requires these comma-separated lists of all the sample data. So here, the input says sample data gut concoct scaffolds bin. Then it says sample data sample human gut max bin. So basically, you specify the file names each individually as a comma-separated list. And then here, you should give a list of the actual binners that you have used. There's two things that are required if you're doing it exactly this way in this Conda environment. One is the default search. I think I mentioned that dusttool uses single copy genes to score each bin. Now, the default search engine for finding that is usearch. Usearch is proprietary software, and it cannot be downloaded through Conda. So basically, if you try to run it this way, it will tell you can't find usearch, make sure usearch is installed. So if you have usearch, you can download it for free in academic setting. It's great software. You can just make sure that it's in your path, and then you're good. However, it does give the option to use different search engines. And we will use the Diamond search engine because that comes with some of the binning software in this environment. So the nice thing about using this Conda environment is that the entire thing is fully self-contained, and you can run the whole process, all the individual binners, plus dusttool without needing to import extra software. So we use search engine Diamond. For this purpose, I'm going to crank up the threads a little bit. This is not what you can do on your own computer, but if you have big data, you can't run this on a laptop, unfortunately. And then the last thing is this write bins flag. The write bins flag gives you, if you don't use the write bins flag, it just gives you a list. Whereas if you do, so if you set that to 0, which is the default, it just gives you a list of which contigs belong in which bins. But if you actually want to work with your bins, you need the FASTA files. So write bins 1 gives you the FASTA files of your bins. And then you just run it. And as you've given this FASTA files, what it first will do is will first annotate your contigs, which means it will call genes. This is covered as part of the functional annotation group, but it will run the program protocol. Now it will run for a little bit, and then once protocol is run, it will use the program Diamond to compare. I defined the single copy genes that it looks for, the marker genes. Once those are found and detected, oh, there we go. It now has used it's finished predicting the genes. Now it will identify the single copy genes using Diamond, calculate the length of all the contigs so that you have an overview. And now the actual DAS tool is running. It started with 20 different bins, and now it decided that 15 of those 20 bins were good. And the sample will be in sample output ls. So if we look at what we have the README files for our different bins. We have an overview of the steps that I briefly showed a few times. We have the sample data that we looked in, and then here we have sample output. It's important to know for running DAS tool that sample output is a required directory. You have to make that before you run the tool. It will not make the directory for you. And as we see here, that is specified with the sample output DAS tool run 1. So your O flag gives you the prefix that all the files will be called. You'll see the DAS tool prints a bunch of different outputs, and the ones we'll be most interested in is the bins over here. So the DAS tool run 1 DAS tool files give you anything that is done in the DAS tool file. The .eval files tell you how good each individual binning tool, each individual bin of each individual binning tool worked. Then the proteins are the ones that are predicted with protocol. And then the archaeal single copy genes and the bacterial single copy genes are shown here. So if we look at sample output, and then look at our DAS tool bins directory, what we have here is we have the 15 bins that it has decided were valuable bins to take. So now we have concoct 17, 77, 72. We have 3 concoct bins, 4 max bin bins, 4 Metabut bins, and 4 tetraes bins. I'm sure the sample data was selected in such a way that yes, it would take all of the individual ones, or it would take input from all the individual binners, but it also shows you how useful it is to take a binning aggregator and put all the things together. This is where I'd like to leave it today. So I'm going to stop screen sharing, and I'll provide this, the way to set up this Conda environment and the tool as a binder on the BVCN website soon.